I'm starting the recording right now.
Okay, you started? Okay.
Started the recording, and now I'm going to make you the host.
Okay.
I haven't done that yet, sorry. For some reason, you've disappeared from my participant list. I'm not sure why that is.
Oh.
It's odd. Are you the host right now?
Let's see. It doesn't say so, no.
Okay, so let me just, you're just not showing up in the list. Okay, there you are. And so we'll make you the host. And so you are now the host.
Okay.
And did that transfer? Did you see an announcement that you're the host?
Yes, I
Okay,
did.
perfect. Okay, so let's get started. So welcome everyone. Good morning, good afternoon, and good evening. There's people from all over the place, so depending on where you're viewing from, welcome to our monthly Dementia Journal Club, broadcasted to you live today from the St. Boniface Hospital Research Center. My name is Ben Albensi, and I'm the host for this journal club. Today, it's really my distinct pleasure to introduce Dr. Brian Balin. Dr. Balin completed his PhD at the University of Maryland School of Medicine in Baltimore. He received postdoctoral training at the University of Pennsylvania School of Medicine in Philadelphia. And currently he's in the Department of Biomedical Sciences at the Philadelphia College of Osteopathic Medicine. He's a professor of neuroscience and neuropathology. He's also the director of the Center for Chronic Disorders of Aging and Basic Sciences. And he's also the chair of the Department of Biomedical Sciences. So Dr. Balin is really internationally recognized expert in Alzheimer's disease. I don't have his CV in front of me, but he's highly published in peer-reviewed journals, and he's written a number of reviews and book chapters and experimental papers, of course, on the pathology of Alzheimer's, in particular on the pathogen hypothesis of Alzheimer's, and that's what he's going to be talking to you about today. So without further ado, welcome Dr. Balin. And Dr. Balin has asked for me to ask you to hold your questions to the very end. So over to you.
Okay, thank you, Dr. Albensi. I appreciate your invitation. I look forward to presenting today on some interesting information we have that we've garnered over many years now, a couple of decades, in fact. I'm first going to give you a slide on learning objectives of what I'm basically going to talk about today, with defining the pathogen hypothesis and discussing epidemiological features of Alzheimer's, along with classification, signs, and risk factors, and describing these pathological entities and how they relate really to neuroinflammation and infectious insult that we're really studying quite a lot in my laboratory. And this perspective on Chlamydia pneumoniae, which we believe is involved with AD neuropathogenesis. So as I go through, we'll start to see how that unfolds, and hopefully I can at least inform you of how this particular pathogen is involved, we believe, with a lot of the cases of sporadic disease. So what is the pathogen hypothesis? Well, in essence, it's what's up here on the slide, with the pathogenic microbes either getting into the brain or even present in our bodies systemically, can activate a response, typically inflammation in our bodies, that can then coincide with the development of neurodegenerative disease, where you have things such as beta amyloid and tau neurofibrillary tangle depositions occurring in brain tissues of people that develop this disease. So the first thing I wanted to mention is that pathogen suspects that have been looked at and are being looked at currently. And you see here viruses, bacteria, fungi, and parasites. And many of these organisms have shown up in Alzheimer's disease I'm admitting all, first of all, have shown up in Alzheimer's disease through different types of analysis. Often it's either through PCRing, through immunohistochemistry, through culturing. So some level of analysis has been done to identify these types of organisms. I'm going to focus on the Chlamydia pneumoniae organism that we found years ago, and I'll sort of describe how I came about doing that. But first, with Alzheimer's disease, it is a global epidemic and an economic crisis. As you see here in this chart, by the year 2050, we're talking about a massive amount of people that would actually go on to develop Alzheimer's disease. A lot of these individuals are of the older age group. So as we're living older, we're running into now an issue where a large percentage of individuals 85 years and older are actually developing this disease, and it is worldwide. Currently, we have a significant proportion of individuals that have Alzheimer's disease. The current tally, in essence, is about 50 million individuals worldwide. Different places have different numbers. Here in the States, we have about 5.8 million, maybe up to 6 million at this point in time. In Canada, there's approximately 750,000 individuals with Alzheimer's disease currently. So we know that this is something that is really affecting multiple populations, and it's leading to an economic problem as well. The estimates are that this could be in the trillions of dollars spent on a yearly basis, taking care of the people that develop this type of dementia. And by the way, this is one type of dementia, and there are many other types of dementias as well, that as we lump them in, these numbers of 50 million actually go much higher. So the classification of Alzheimer's disease, there are basically two forms. There's a sporadic form, which is the one that we've been studying, is about 95% of all the individuals that develop Alzheimer's disease worldwide have this form. It's one of that has its greatest risk factor with age. Typically, we look at 65 years of age and older, that population, and it's considered to be a late onset form. Although even with late onset, it may start developing years, potentially decades prior to seeing the manifestation of the disease. And then you have the familial form of Alzheimer's, which is about 5% of the entire population of people developing Alzheimer's. And this is really due to genetic mutations of different genes. And as such, those individuals will develop the disease as early as their 30s and into their 40s, early 50s. And now that's obviously a problem, because you're seeing this younger generation or younger individuals developing this at such an early age. So the major risk factors, again, age is the #1 risk factor. And again, the genetics, especially for the familial form of Alzheimer's disease, where you have the genetic mutations. I must tell you, though, that there are other genetic issues that are not mutations, but where you're seeing that through GWAS analysis, there are other genes that have been shown to be involved with Alzheimer's disease. One of the key ones is the apolipoprotein E epsilon 4 apolipoprotein type. And when you have a single a gene expressed with ApoE4. It's more of a polymorphism than it is for any type of mutation here. But if you have that expression of ApoE epsilon 4, a single expression would give you about a 3 times risk of developing Alzheimer's disease as you age. A homozygous expression, where you now have both genes with ApoE4 expressed or present, leads to approximately an 8 to 11 times increased risk. So that is a key genetic risk factor, even with sporadic disease. The other risk factors listed here are atherosclerosis, diabetes, head injury, toxins to some extent, and infection. And all of these different ingredients have been looked at and are currently being looked at as well as how they actually are involved with the development of this type of problem. Seem to be stuck here. Okay. Alzheimer's disease versus typical age-related changes. I wanted to present this because I'm often asked, well, what is the difference here between just normal aging and really an Alzheimer's aged individual and an Alzheimer's diseased individual? And here's just a little type of table here demonstrating the difference being, for instance, with Alzheimer's dementia, it's very severe on the left-hand side here of things of having a conversation, of cognitive deficit, of memory issues that are severe, of poor executive functioning, of poor ability to have activities of daily living and carry those out, versus things that are more transient, where you have now bad decisions maybe made once in a while, forgetting something, but then remembering, etc. When you look at the brains of people with Alzheimer's disease versus those that would be normal as a aged matched type of process here, you see on the left with the normal, the lateral view at the top of the slide of the frontal, parietal, temporal cortices and cerebellum, etc. And then the coronal section. And the gyri are nice and compact together. The sulci, you can't see the separation between the gyri very well at all. And if you look at the bottom part of the coronal section, yeah, I think maybe I can point it out here. I don't know if you see the arrow or not, but you see the hippocampus is very well intact, even in an older individual. Over here with AD, you see now the gyri are shrunken. You literally are losing mass of brain tissue. And the sulci are the widening areas between those gyri. And then down below with the coronal section, you can actually see that you have hydrocephalus ex vacuo, it's called, where enlargement of the ventricular system in the brain, and also basically atrophy to a huge extent of the temporal cortex, especially of the hippocampal formation, which would be right in where my arrow is pointing. So the disease is named for Alois Alzheimer. He was a psychiatrist with the microscope. And what he saw with silver staining of brain tissues of some demented individuals that came in to see him and then went on to die, and at autopsy, staining of the tissue with silver stains, which was the common practice at the time, you could see accumulations of plaques and tangles. And typically these plaques were extracellular and the tangles were actually sitting in dense deposits inside of the nerve cells. We now know those to be the neurofibrillary tangles and neuritic senile plaques. And while I'm talking about these two main pathologies in Alzheimer's disease, you must realize that they're not the only pathologies that can show up in this particular disease. You may have other types of pathology as well. In particular, you could have quite a lot of inflammation of the glial cells responding in this disease, both astroglia and microglial cells. You can also have systemic immune cells that will show up in Alzheimer brain tissues. You can also have other types of deposits like alpha-synuclein, TDP43. These are other ingredients that might show up in different cases of Alzheimer's disease. But let's focus on these particular components. You have paired helical filaments, which are made up of hyperphosphorylated tau proteins. These can form inside of now your neurons, which there's an arrow here pointing to the neuron containing that, and also in neurites that are now dystrophic, meaning these are the processes of the nerve cells that are now damaged or atrophying. The nerve cell is atrophying. And these pieces of material are present there inside of these atrophying pieces of the nerve cell. And that's what we call these paired helical filaments. Neuritic senile plaques down below are really made of now these dystrophic neurites, and now surrounding them is this extracellular material, this amyloid, which can take on a number of different forms. The A beta 1 to 42 form, and I'll go into this a little bit as we go forward, actually is the main component of these established plaques that accumulate in Alzheimer brain tissues in an extracellular fashion. So what causes sporadic Alzheimer's disease? This is an early diagram from one of my graduate students in the late 1990s. And Jamie Arkin came up with this. And this was like a pathway that we were looking at, or pathways, just at that time. And now the complexity is enormous. And anybody studying this disease realizes this, that there's no single component here that is independent of any other component. And because of that, you now have to look at this as being a very intertwined, three-dimensional type of process that's happening with all of these different pathways involved. So I'm going to try to break down some of this based on the infectious components that we've been looking at. So what initiates the neuritic senile plaques and deposits in sporadic Alzheimer's disease? Well, this amyloid cascade hypothesis, where you get these amyloid plaques, has been the prevailing hypothesis for decades. And this is where most of the drug development has actually been directed. And unfortunately, even more recently with Biogen's drug, these drugs seem to fail. They're not working when you go into the human population, human trials, of either blocking amyloid from accumulating or trying to get rid of the amyloid that has accumulated from brain tissues. The other major hypothesis that most people accept now is neuroinflammation occurring in this particular disease. And it's intriguing to think that from the 1990s to basically the late 1990s, the people studying and defining neuroinflammation were actually struggling to convince the field that inflammation in the brain was a key element to the development of this disease. Now it is widely accepted, though, that inflammation is a key element. And the tie-in here seems to be that inflammation may be generated through the secretion or this abnormal accumulation of amyloid. We happen to believe that inflammation actually may be occurring prior to amyloid being deposited and being made and secreted out of cells. And that's sort of now a debate in the Alzheimer world at this point in time. So let's go back to amyloid a moment. Here we have amyloid is derived from the amyloid precursor protein and that particular gene. You have a transmembrane protein, a type 1 transmembrane protein, and it can be cleaved by different enzymes. So you're going to have alpha secretases, beta secretases, or gamma secretases. Now, as a type 1 transmembrane protein, it's only going through the membrane one single time. And the region I'm pointing out here with the arrow is this A beta region, is the critical region that actually is leading to the deposition of amyloid over time. It can be cut by these different secretases in different places. If it's cut in the middle of that orange segment that is in the slide, you will not lead to amyloid accumulations. It will not be a generation of the ability to lead to plaques eventually. So you don't get accumulations of amyloid. However, if you get beta secretase and gamma secretase cleavages, you can cleave out this segment of this orange segment of beta amyloid that actually does deposit over time. What's interesting here is that the cleavage of the enzyme by these enzymes leads to the initial accumulation of amyloid in an intracellular fashion, and then it gets secreted outside of the cell, where it will form into multiple peptides, into oligomers, and into polymers over time, and then into the more of the macromolecular neuritic senile plaque that you see in the disease. So this is what we think is happening. Now, in the genetic form of Alzheimer's disease, that familial form, there are different mutations in genes that code for beta secretase, gamma secretase, and also for the amyloid precursor protein gene, okay, in those genes. So those mutations can lead to, in familial Alzheimer's disease, increased production of amyloid. But that's not happening in sporadic disease. Those enzymes and those proteins are not mutated in sporadic disease, but you still get an increase in amyloid. So how is it actually happening? So here's where we think the merging of the hypotheses needs to be considered with these two major hypotheses. Now, you must understand too that these aren't the only hypotheses that people have for Alzheimer's disease, but I think these are key in trying to understand what's happening in the disease process, especially for sporadic Alzheimer's disease. And the crossover here to us is this infection hypothesis. That we are not by far and away the first people to think about infection as being involved with Alzheimer's disease. In fact, if you think back and look back into history, Oscar Fischer, who was a contemporary of Alzheimer's in the early 1900s, actually was considering that different types of actinomyces organisms might actually be involved with the generation of Alzheimer's disease, thinking that an infectious component actually was there. But it was not until now going forward in time to really the late 1980s until people came back to infection as being a serious issue in this disease process and starting to uncover data of different types of infectious agents that would be involved in this disease. And we're still at that point where we're still trying to understand how are these infectious agents involved and how could they actually cause the problem, not just be associated with a disease entity, but actually cause the problem. So this leads us to the chlamydia pneumonia issue that we've been studying. Now, before I talk about the organism itself, how I stumbled into this actually was with Alan Hudson. He and I were at the same institution. We had a talk one day, and we were talking about how this particular organism was being reported on with atherosclerosis. And why did we bring up this organism? Well, Dr. Hudson is a geneticist that historically has studied chlamydia organisms. And typically he was studying Chlamydia trachomatis, and he was studying that in the aspects of reactive arthritis. But he had knowledge of this organism now coming about. This was first defined in 1987 by the Grayston group out of the University of Washington. And it was an isolate from a respiratory infection. And they defined it as Chlamydia pneumoniae. So when Alan Hudson mentioned atherosclerosis to me, I jumped at that and said, do you know if anyone's looked at this in the brain? And I said that because my background with my PhD work was on blood-brain barrier. And I always had thought that with Alzheimer's disease, which was also my background with my postdoc, that atherosclerosis was a linkage to the particular disease process of Alzheimer's disease. So it was natural for me to think that way. So we decided to look in our brain tissues. So I'm going to get to that in a moment. So what is Chlamydia pneumoniae? It's a common respiratory pathogen. It's involved with about 20% of community-acquired pneumonias. So it is a community-acquired issue. It appears that in many cases, it goes unnoticed. People don't know they've been infected with the organism. This is involved with also asthma, chronic obstructive pulmonary disease, other types of respiratory ailments. It's also been found with coronary artery disease and atherosclerosis. It typically can get into the bloodstream through inhalation into the lungs. I'm going to talk more about the pathways in a moment. But once it's in the lungs, it can be picked up by monocytes and trafficked throughout the vascular tree, and it's thought that that's how it can be involved with atherosclerosis. It's an obligate gram-negative intracellular organism. Once it infects us inside of our cells, it grows in vacuoles, typically inside of cells. And just about all cell types can be infected, by the way. There is serological evidence in the elderly. As we age, we have now found that a significant proportion of older individuals show that they've been infected through serology. And there you can find IgA, IgG serology positivity. Admit. Okay. So what did we find? So after I had the discussion with Dr. Hudson, I went back to my laboratory and pulled out frozen brain samples from my -80 freezer. And over the years, we've collected Alzheimer brains and normal brains, et cetera, and other neurological diseased brains. And I took out material from different brains and just took pieces and told him, okay, he was an expert geneticist looking at chlamydia organisms. I'm going to send you this particular material, see what you can find in it, if there's anything there at all. And he did PCRing of this material in a blinded fashion. So I blinded the samples. He didn't know what was normal or what was Alzheimer's. And he came back with positives for Alzheimer's and negative for the controls. And we did it a second time. And the second time I used different tissues. He came back with the same data. We said, we have something here. Let's follow this. And to this day, we're still following this to understand what is this organism doing in us, potentially in relationship to Alzheimer's disease and maybe other diseases. In essence, here's a summary of what we found. 17 of 1980 brains positive by PCR, 1 of 19 age-matched control brains. It was present when we looked in the brains itself. We saw the organism by immunohistochemistry. It's present in areas of AD neuropathology. You could see plaques and tangles nearby. You could see the label for infected cells. We had glial cells and neurons were infected. And here's a picture at the bottom here of this brown material. It's labeled for the chlamydia inside of a nerve cell. This is a big vacuole containing it. Over here on the right-hand side is an electron micrograph showing the two forms of the organism, the elementary body forms, which are these dark forms, and these more translucent forms, which are the reticulate body forms. These are the more metabolically active forms. The dark forms are the infectious form. There are also in here, over here and over here, forms that we would term are intermediate bodies, which would basically indicate to us that there is a level of persistence undergoing in these cells, in these chlamydia organisms that are sitting inside of a cell from a human Alzheimer brain. And again, we used multiple techniques. So we didn't want to rely on only one technique to identify what we have here. So we used multiple techniques to actually identify what was present in the brain. And here you see a lot of electron micrographs, A through G, listed here. These are showing the depictions of the organism that we saw in human brain. This one is a pear-shaped organism in B that is very typical of Chlamydia pneumoniae. And down below, we have now these little particles, gold particles, in F and G that are encircling the organism. These are representative of antibodies that had gold tags on them. And this is a process called immunoelectron microscopy, where you could actually now label for the organism this way. So what we did was we wanted to use all these different techniques: immunohistochemistry, electron microscopy, the immuno-EM, PCRing.
I had to re-download it, Mom.
Later on, I
literally deleted the entire program on my computer and then re-downloaded it.
Here's somebody else out there.
But I have no idea why that happened.
I think somebody has their
volume. I logged in and I tried to enter the
Excuse me, Courtney. Courtney, please turn your mic off. The rest of the audience can hear you. Thank you.
So what's interesting here is that in regard to the EM, I wanted to focus on that a moment, is that historically, going back into the 60s, a lot of electron microscopy has been done on Alzheimer brain tissues. And in the 80s and 90s, a lot of focus was on lysosomes and how lysosomes actually were present inside and sort of now providing a nidus of lysosomal activity inside of a lot of nerve cells. And if you don't have if you have an eye for what you see here, when you see these dense bodies here, these look like they could be lysosomes, actually, inside of a cell. And in essence, I actually think this is what's been missed by looking at the electron microscopy, is that if you didn't know there was potentially an intracellular organism present in these brain tissues, you would not know to consider that what you see here is not of the lysosomal variety or of accumulations of dense bodies that are typically seen in cell biology by electron microscopy. So if you didn't know that this could also be something else, you wouldn't even think that this was something foreign. And this is where we have now diverged from a lot of the people that looked at electron microscopy to now reconsidering what is it that you actually are seeing. Anyway, when you do the immunohistochemistry and cell culturing and looking for cells that are infected, you can see through immunofluorescence, these yellow accumulations here are representative of the chlamydia organism inside of cells. Tissue culturing. In order to have this, prior to publication of our first study, we actually grew the organism out of human brain tissues, and these are in monocytes in culture. And so these are the light micrographs up top here with A, B, and C, and the organisms within the cytoplasm, and the EMs of those cells down below in D and F, showing the organisms inside the vacuoles, inside the cytoplasm. So additional human studies have been done. Glial cells, neurons, endothelial cells have been found to be infected. The brain areas, we believe truly that this is an olfactory infection. So the olfactory neuroepithelial cells, principally, we think are being infected by this respiratory organism. And that once it's inside there, it can get into the brain through our olfactory centers and go right into the limbic system, which is our entorhinal cortex, our amygdala, and then farther in then into, from the entorhinal cortex into the hippocampal formation. So this is a real interesting scenario, and thinking that a respiratory organism could do this and set up the stage, we believe, for inflammation. Also, we think the blood-brain barrier is involved in this infection, and we have evidence in culture or in vitro to actually suggest that. Chlamydia pneumonia detected in blood samples from AD and MCI patients. MCI is mild cognitive impairment. This is a study that we're currently working on writing up, but this is some data from that suggesting that in people with mild cognitive impairment and that have been diagnosed with AD, now this is clinically diagnosed, that if you look at their white blood cells by doing a Buffy code analysis of their blood and look in their white blood cells, you can actually see elementary bodies, intermediate bodies, and reticulate bodies. And these graphs are depicting now those different status of the organism. So literally, you can do the analysis of your white blood cells to determine that you have the intracellular organism on board. Now, what I'm not showing you here is that we also did immuno-EM on those particular cells, and you can label for the organisms with antibodies specific for Chlamydia pneumoniae for those particular organisms. So the pathway for proving pathogen involvement in Alzheimer's disease, in general, this could be applied to almost any pathogen. But what we found is that we first found it in human AD brains. And that was basically by just looking and having probes to be able to do that. But then you can now start to study it in animal models and cell culture, and then start to evaluate, what are you seeing? What is the organism doing? How is it associated with pathology? And then what's happening inside those cells? What are the pathways that are being activated? How are the cells responding? And how does that correlate to what we see in Alzheimer's disease, even independent of an infectious process? So here we have routes of infection into the brain, the olfactory route, as well as now inhalation into lungs, where the monocytes can pick it up and get into the bloodstream. And we think ultimately you're going to get generation of these amyloid plaques as a response to the presence of this organism. But the amyloid, we think, is also is being produced by the infected cells. So we have a process by which amyloid can show up when you have infection on board. And then if amyloid starts to show up and be secreted from the cells, your glial cells will respond and also now start to create inflammation that can now lead to more damage in the nervous system and possibly more amyloid generated. Sniffing out Alzheimer's disease, this is something where it's been recognized that the inability to recognize smells and combined with the lack of awareness of the impairment of odor perception shows this impending Alzheimer's. And Devanan actually published this in 2000. He's been looking at olfaction for some time. Olfaction also has been noted since the late 1980s to be an issue with a lot of the neurodegenerative processes, including Alzheimer's and Parkinson's disease also, and other diseases such as schizophrenia, for instance. So it's an early impairment, we think. And that's where the respiratory infectious agent, early impairment of olfactory neuroepithelial cells that are exposed to this agent, that type of connection is where we're really focused on trying to understand that as being a precursor then to now what's happening in the limbic system in the brain proper. So here's an animal model. We developed the animal models by intranasally infecting animals with Chlamydia pneumoniae, either that was obtained from our brain cultures of human Alzheimer brains, and or using a laboratory strain of Chlamydia pneumoniae that was originally obtained from a respiratory culture of chlamydia. And here we see that intranasal, the nasal epithelium can be infected if you do the intranasal inoculation. So this is the brown is indicative of antigens for the organism. Down here with olfactory bulbs and the bulbs proper of an animal, you can now find the organism by electron microscopy in vacuoles sitting in the cells in the olfactory bulbs. Later on, you can find actually A beta 1 to 42 like amyloid plaques that are present And not to a degree that you would see with a transgenic animal, obviously, but in this infection model, they're starting to show up in different regions of the brain. And we focused in on the hippocampal region and amygdala, perirhinal cortex, et cetera, where the plaques showed up. By the way, amyloid here in a mouse brain is about 94% homologous to human amyloid. And this stimulation of a normal mouse now with this organism, we think has led to now the production of this mouse amyloid showing up in these brains after infection. And we used antibodies actually that were made to human amyloid to label for these amyloid deposits. So you also see a glial response with astrocyte activation. Over here, again, more amyloid deposition with the brown spots that you see in these micrographs. So in some other studies that we've done, we've looked at now, considered the following: location of the organism once we have infection in a mouse, what strains of organism were being used for the infection, the dosing, the length of infection, and the age of the animal. So in this slide, the two slides here, two graphs that we have, the upper and the lower down here, we're looking at now location. Oops, let's go back. We're looking at location of the organism over periods of time. And you can see over a 4-month period of time, you can find plenty of organism present in the olfactory bulbs. What's interesting here, depending on the organism, we may not see it in brain proper. It's residing in the olfactory bulbs over a length of time, but it may not be in brain proper. And we found that the respiratory strain sort of gets eliminated over a period of time, but the brain strain of the organism actually could remain. The other thing we saw here is, if we go down to this particular graph, is the dosing. So if you have low dose versus high dose, low dose was 5 × 10⁴ organisms, high dose was 5 × 10⁵ organism. Both ways they were delivered intranasally, just dropped into the nares of the animal. One dosing is all this was for either low or high doses. And you'll see that down here on the left-hand part of the slide is that with low dose, at 14 days post-infection, you don't really see any evidence of the organism in this animal. At high dose, at 28 days post-infection with low dose, you'll start to see, even in young and old animals, you'll see evidence of infection. With the high dosing, which is 5 × 10⁵, at 14 days and at 28 days, 14 days these two, 28 days these two, you'll see evidence of the organism in all of those animals. The animals here were 6 months for young animals, 6 months of age, old animals, 20 months of age. And if you see on both of these graphs, I'm pointing out on the left-hand side here, the higher level, and over here, the higher level. There's more organism that's maintained in the older brains than in the younger brains, suggesting then how age could be a major factor here of maintaining this type of infection in the brains of these animals, and giving us a rationale of why age could be a big risk factor. Okay, so let's move on. So cellular mechanisms, very quickly, I have different mechanisms that we've studied and published on. Here's a mechanism of looking at an in vitro blood-brain barrier. In essence, what this tells us is this, is that in doing this blood-brain barrier study, if you have infected human brain microvascular endothelial cells and infected monocytes that are now put together, the monocytes can traverse between the junctional complexes of your human brain microvascular endothelial cells. And they can do that because the junctional complexes are transiently affected with infection, meaning they actually will change over a period of 72 hours. So within post-infection, up to 72 hours post-infection with an endothelial cell, the junctional complexes that have formed actually now demonstrate that you get an upregulation of the adhesion molecules, but a downregulation of the tight junctional molecules. The occludins proteins is downregulated, meaning the junction then can still be leaky because you don't have a tight junction formed between them. When you put monocytes on top of these cells, the monocytes will migrate through, and in this particular model, you could see that. And what's depicted here now in this blue down in here are monocytes that have come through this endothelial barrier. And it's because the monocytes actually now express, that are infected, they express integrin molecules that, just like with inflammation, any inflammation, they allow you to bind to the endothelial cell and crawl through the junctional complex. So these studies that we did here demonstrated a proof of concept that infected monocytes circulating in the bloodstream could actually traverse a blood-brain barrier in theory in an animal or a human. The other things that have been looked at, autophagy is initiated, apoptosis is inhibited. Autophagy often is initiated when you have an intracellular infection. It's trying to clear the infection. We noticed it was initiated and you get a lot of lysosomal activity, a lot of endosomal activity. However, you do not get binding of lysosomes to the phagosome containing the Chlamydia pneumoniae. And because you don't get that, the organism resides inside the cell and can now control how the cell is going to respond. And basically, apoptosis is inhibited, BCL2 is activated, so it's anti-apoptotic, and some other aspects and proteins are actually activated that are anti-apoptotic, so that now you have a long-lived infection in these particular cells. And these experiments were demonstrating those activities. Calcium dysregulation is another activity that we find is dysregulated. And we find that when we do have infected astrocytes in culture, S100 beta is a protein that gets elevated enormously. And that S100 beta has been seen in Alzheimer's disease too to be elevated, and it's thought that that's involved with calcium regulation. If calcium now is increases its presence inside of the cells, we think you're getting the phospholipases activated. You're getting enzymes activated too that are disrupting cytoskeletal processes and proteins. And we think that's part of the process here, that chlamydia is stimulating this to occur, so that now you're impacting how cells are going to function. The other interesting thing we see is inflammasome activation. And what's interesting is that sort of correlates with the autophagy and apoptosis. If autophagy doesn't go to completion and can be actually inhibited at some level, inflammasome activation can actually hike up. And we see with the chlamydial infections that inflammasome activation is hiked up. There are many different types of inflammasomes. Typically, they're activated in cells through the toll-like receptors and activation that way. We find with chlamydial infections, it's the AIM2 inflammasome in particular that gets activated. And that's an inflammasome that's located, it's a complex of proteins inside of the cells that actually gets activated to activate your NF-kappa B transcription factor to now produce, stimulating the production of IL-1 beta and IL-18, cytokines that are pro-inflammatory. And we have found that the AIM2 inflammasome recognizes cellular cytoplasmic DNA that's foreign to the cell. So it suggests to us that even though the chlamydia seems to be in a vacuole, at times there's a level of processing of this organism to allow for some DNA to escape the vacuole, to get into the cytoplasm, we think, to activate this particular type of inflammasome, which is very intriguing to us, and we still have a lot of studies to do on that. But it correlates with what's also been seen with Alzheimer's disease, with activation of inflammasomes leading to inflammation and increased inflammation and damage. Here's another study that also demonstrated that if you infect monocytes in culture, that they will produce over a 5-day period of time IL-1 beta, IL-6, and IL-8. Again, pro-inflammatories that all of them have been associated with Alzheimer's disease and how you would get now increased neuroinflammation generated in that disease process. I wanted to mention the astrocyte again for a reason here, and that is because we have a study that was published last year actually, of infection of astrocytes with chlamydia pneumonia and what was the response. And astrocytes are a major glial cell in the brain. Obviously, they're supplying nutrients to neurons, but they can respond to infectious agents as well that are getting into the brain. And we found that astrocytes can be infected with chlamydia pneumoniae in human brain tissues. So we wanted to look and see what was happening inside there. So in culture now, if you now take infection of the astrocytes in culture, as you see on the right-hand side of the slide, at 48 hours post-infection, you can actually see that you can increase intracellular A beta 1 to 42, which is the red little dots you see inside of these cells. So the intracellular accumulation initially of A beta 1 to 42 in infected astrocytes. What we also see is that if you look at gene expression, what's interesting here is at 48 hours, when you're comparing uninfected, which is in the blue, to the infected in the gold, you'll see that 48 hours, APP gene expression, the amyloid precursor protein from which the A beta peptide is derived, actually is decreased in its expression. So we're actually decreasing that expression. But at the same time, we're increasing expression of one of the genes and its protein that would lead to cleavage of the A beta portion of the APP protein, suggesting that you're downregulating the main gene and the main production of the large protein, but you're cleaving out the existing, or potentially cleaving out the existing peptide of the existing transmembrane protein in the different membranes within a particular infected cell. And here you see that if you measure for secretase activity as well, the pro-amyloidogenic secretase activity, the base 1 activity, is also increased. So not only is the gene increased in expression, the activity is increased. Over here, you see there's a decrease in the non-amyloidogenic. So the ADAM10 activity, the one that would cleave the beta peptide like in the middle of the peptide so it wouldn't accumulate, is not being expressed highly. It's actually downregulated. So this goes then to looking at the secretases, base 1 here on the left and presenilin 1 on the right. And the red that's labeled in those cells is actually representing the base on the left and the presenilin on the right of the infected cells. And if you do a fold change analysis, you can see that it's significantly different at 72 hours of base 1 and with presenilin 1, suggesting then that with increased protein expression, you're actually now cleaving out the beta peptide. So infected cells are promoting, in astrocytes in this case, the production of amyloid beta, the 1 to 42 form, which is really the damaging form in the disease. So overall, this is what we think is happening with chlamydial infection of cells leading to amyloid processing, which eventually leads to degeneration. But we have a few other things to look at. Here's a comparison of Alzheimer's disease to chlamydia pneumonia infection. And everything over on the left here is what has been seen with Alzheimer's disease, all the different types of processes and how they now are involved with the disease process. On the right here is chlamydia pneumonia infection, and I have arrows drawn between because they correlate quite well with what we've observed with the dysfunctions we've seen with Alzheimer's disease. Now, in essence, this is a correlation. It doesn't prove causation at this point in time. But we're gathering evidence that is now becoming overwhelming that we have crossover of pathways where chlamydia pneumonia infection actually crosses into what was recognized in Alzheimer's disease without actually even considering that you have an infectious agent on board. That's why we think this is so important and such an important find, and why we're focused on trying to understand how this could be truly causative in late onset disease. Other features that are observed, the other risk factors and chlamydia pneumonia infection. I really haven't gone into this with the ApoE epsilon 4, but there are publications that we have of this relationship with chlamydia pneumonia infection as well. And possibly the ApoE epsilon 4 is helping the organism infect an individual, at least individual cells, and maybe individuals that express the epsilon 4 genotype. Then with atherosclerosis, we have a linkage, diabetes, and neurotrauma. So all of these things are risk factors for the overwhelming issue of Alzheimer's disease, and all of them could involve infectious agents, and in particular the chlamydia pneumonia agent, but maybe all infectious agents down here for instance, neurotrauma. So it comes down to that we may have to look at the problem with infectious agents as which ones are primary, which ones are secondary, which ones could occur together in a number of cases with this particular disease. Here is just a schematic showing infection invasion, cellular dysfunction, and neuroinflammation. In essence, a summary of what I've just talked about, showing how they're interrelated and would lead to a degenerative state in Alzheimer's disease. And this is my group over here on the left. The upper left, we were at a science festival on the Parkway in Philadelphia, which is held every year by the Franklin Institute. And we rented this giant brain that's out. It's a walk-through brain that was out there. It was a big hit for all the people that were actually coming to this festival. Here's the Philly Fanatic. If everybody knows the Philadelphia Phillies has a mascot, that's what that individual is there. And then I just wanted to recognize the contributions that so many people have made here. Our funding sources, our collaborations, the student contributions, which is enormous. And this couldn't be done by one person. It has to be done by a whole group, a whole village. And this is the last slide I wanted to show. And that's discovery consists of seeing what everybody has seen and thinking what nobody has thought by Albert and Georgi. And I love this slide because it's so true. Science is still done through serendipity. And basically that's how we have gotten into the chlamydia pneumonia and Alzheimer world. So thank you very much. I'll take any questions if we have time for that.
That's great. Thank you very much, Dr. Balin. So a very comprehensive and interesting talk. So let me start off the round of questions. We have a couple of minutes. I guess you probably get this asked again frequently. I guess it's a challenge when studying infectious disease and Alzheimer's is to try to really pinpoint the smoking gun, as it were. And what I mean by that is that I guess what's confusing to me is the time course. And so I guess it's possible that someone could be infected like 15, 20 years ago, but that now only 20 years later are you looking at samples or other markers to evaluate Alzheimer's disease. So I guess there's an important disconnect there that makes it more challenging. Is that true?
There is, and I would agree with that. I think that this organism presents some very interesting issues for us. Even very young people can be infected with this, but we're not seeing that there's brain damage or there's some type of cognitive change in those individuals. And usually that goes through middle age as well. And so we've considered that it's an immune response that we typically have that is maintaining the ability of us to stay healthy. Now, the organism still may be on board. It still may have gotten access into the brain. We don't know that for sure. We do know there have been a few studies looking at individuals that had atherosclerosis that died at a relatively young age, about 60 years of age on average. And a couple studies came out of Italy And they showed that they could find Chlamydia pneumoniae in brain tissues of those individuals, but they didn't see damage, which is very interesting. So it may be that it has to be there and reside for a period of time, and then you can go on to develop the disease process. So that it seems that we're still not sure when this is happening, but it is a conundrum because to figure that out, we have to do major population studies and follow them for years and years at a time to really try to understand, okay, how did this get there? Was it there? One of the studies that I would like to do is analysis of olfactory tissues. Maybe with the COVID issue, we might be able to link into something there where evaluating olfactory tissues, even for other agents and multiple agents, and chlamydia being one of them, we could spot when this is on board. And if it's correlating then to cognitive change. And I think it actually does, but again, it requires more study there.
Right. Okay, thank you. So other questions? Could you unmute yourself and
so I can ask one question.
Yes.
Yeah, excellent talk, Dr. Brian. I have just a general question. So you said like chlamydia induces amyloid plaques, right? So I was thinking, like, did you check the effect on hyperphosphorylation of tau or any other tau modifications as well?
Okay, so what's interesting here is that in short order, we do not see tau changes. We've seen some enzymatic changes, like a GSK3 beta, which is a glycogen synthase kinase 3 beta, an early enzyme change that is leading to phosphorylation of tau. But we think actually that it's amyloid first and then tau is actually activated to change later. Now, the tau accumulations do correlate to the nerve damage, the nerve cell damage. That's more endpoint, I think. And obviously the synaptic changes are occurring earlier. So I think synaptic changes after amyloid are starting to occur, and then you're activating the cells through amyloid binding to cells as well, which has been shown actually to activate signal transduction, leading to hyperphosphorylation changes of the tau protein. But I think also, if you think about it this way, if you're starting to get any calcium changes as well and channel changes, you may be activating those enzymes that are leading to disruption of microtubules and other cytoskeletal components, sort of like opening the window here of influencing how enzymes are going to act on the tau protein. So
thank you.
That's more downstream, we think, than from where we are with this particular infection.
Thank you.
So we're a little over time. Maybe we have time for one more quick question, if you have it. If you could
Ben, it's Mark Delvidio. Can I ask a question?
Absolutely. Go ahead.
So I'm a neuropathologist, and I don't do dementia research, but I go to the neuropathology meetings annually, and I literally never hear about this stuff. And I'm curious whether research in this area has become balkanized. And I just, the advantage of Zoom meetings is you can read stuff while you're listening. So there's a recent review suggesting that multiple different organisms have been detected in Alzheimer brains, and that it might be, rather than a cause effect, more of a reflection of dysregulation of the immune system in the context of a lifelong exposure to different diseases.
Yes. Addressing the neuropathology part, typically when I present this information, I'm either going to the International Alzheimer's meeting or the Alzheimer's Parkinson's meeting, where we have a forum of a few individuals that present on infection in Alzheimer's disease. It is not widespread, though. The issue about the cause and effect and the immune system, this is where it gets pretty complicated, because you can have activation events, even through the gut microbiota, for instance, and the gut-brain axis, that would lead to cytokine changes, inflammatory molecule changes that it can influence cognition. That can happen. The specificity is where I come into the equation with a respiratory organism. And I say this because of this reason, is that the earliest damage in Alzheimer's disease is the entorhinal cortex, directly connected to olfaction, which then also it's directly connected into the hippocampal formation. And also you always see damage in the amygdala. Now, I'm looking at this as sort of like an Occam's razor type of approach. What is the simplest explanation of why you see specificity of damage in those particular regions of the brain at the earlier points in time of disease evolution? I think it's because of olfaction damage. We'll put it that way. Now, it could be pollution, it could be trauma, it could be infection, it could be a combination of these things. But I think that gives us the specificity of why that area of the brain, even more so than looking at the blood-brain barrier as being the insult area for that particular region. So looking at it that way, once you're in the brain, once something is in the brain through olfaction, and you've impacted the amygdala, by the way, I believe you now open the window to all other areas of the brain being affected. Now, a lot of people are looking at the gut-brain axis as if the vagal nerve is being insulted and things are coming through the vagus into the brainstem. If that's the case, most of our diseases would be starting in the brainstem. And while you can see pathology there, it's typically not where the disease is beginning. And so in this analysis, I'm thinking, well, olfaction, the amygdala, entorhinal cortex, hippocampus, these are areas that are key in our focus.
Thank you.
You're welcome.
Okay, well, thank you again, Dr. Balin. Thanks for your time today. It was really a great talk, very interesting talk, lots of stuff to think about. For those of you that may have joined late, I'm going to post the link, the recording later today once it finalizes. So I just want to thank Dr. Balin again, and thank you all for joining us to our monthly Dementia Journal Club. So thank you. And Dr. Balin, I'm going to take a quick bio break and I'll be back in about 2 or 3 minutes.
Okay, wonderful. Thank you.
Okay, thank you.